Hello everyone, and welcome to today's webinar, SARS-CoV-2-specific B cell detection and therapeutic antibody discovery. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Cristina Conforti Andreoni, Global Product Manager for Cell Culture and Group Leader in Translational Immunology at Miltenyi Biotec. Cristina, you may now begin your presentation.
So this is the agenda of the meeting. I would like to discuss about the role of monitoring cellular immune response in COVID-19 and also the recombinant SARS-CoV-2 proteins that we offer and how they can be used for the B-cell analysis. And finally, I will end with a part on the therapeutic antibody discovery. Let's start with the monitoring of cellular immune response. In general, it is important to study the immune response to viral infection. And indeed, all immune cells can have a role in the disease outcome and clearance. Some, like for example macrophages, might have a negative role in the pathogenesis of diseases such as SARS-CoV-2 infection and COVID-19. And these cells can be studied as target to develop drugs that can be used to treat patients in an acute and severe phase of the disease. Other cells, like lymphocytes, play instead usually a positive role in the defense against the viruses and also in the mounting of immunological memory, which gives the protection from future infection. Of course, the goals of vaccination against SARS-CoV-2, as for any vaccination, it is to develop an immune response and therefore measuring SARS-CoV-2-specific T cells and B cells in vaccinated individuals is a very important step in the evaluation of the efficacy of COVID-19 vaccines. The most important difference between T cells and B cells is that T cells recognize via T cell receptor antigens bound to the MHC of infected cells. On the contrary, B cells, via B cell receptor, can recognize antigens of full pathogens present in the microenvironment without the need of a presentation of an infection. Once the B cells recognize the specific antigen, they develop into plasma cells. And the B cell receptor is basically an antibody that lately, as plasma cells, is released and secreted in the environment and can eliminate the pathogens in several ways. Since the antibodies eliminate the pathogens directly before the cells are infected, this response is sometimes called sterile immunity, and it is usually the target of vaccinations. How do antibodies protect us? The most simple way an antibody can protect us is via neutralization. In the case of SARS-CoV-2, for example, some antibodies recognize the protein spike on the virus surface and block its binding to the ACE2 protein, preventing, therefore, the virus to infect the cells of the body. There are, however, other ways that antibody can use to help to eliminate immune response and to kill pathogens or, for example, tumor cells. One example is the activation of the complement or the recruiting of killer immune cells such as macrophages or NK cells. As we will see later, therapeutic antibodies should be able to induce one or more of these mechanisms, and therefore it is important also to test their functionalities. So we've seen how basically a strong antibody response per se should be able to control and clear a viral infection such as SARS-CoV-2 infection. However, It has been observed in several individuals that the level of antibodies in infected individuals go down after several months after infection. And therefore, these individuals are still susceptible to future reinfection with the virus. However, a recent study has shown and proved that B cells instead persist in the blood of convalescents, even over 8 months after infection. And therefore, the B cells could help to prevent future infections or help the individual to clear the virus quickly without developing of a severe COVID-19. Both roles of B and T cells are therefore important in SARS-CoV-2 monitoring. For these reasons, at Miltenyi Biotec, we have developed specific tools to study these cells. In the following part of the webinar, I will illustrate the tools for SARS-CoV-2-specific B cell detection. However, for T cells, we also have several products and solutions I can recommend in order to study the T cell response to SARS-CoV-2. You can find more of this information in upcoming webinars and also in our website. So let's look now at how to monitor antigen-specific B cells using the SARS-CoV-2 proteins. So in the past 12 months at Miltenyi Biotec, we have developed several recombinant proteins of the SARS-CoV-2, as you can see here. All proteins are manufactured in a recombinant eukaryotic system with or without a covalently bound biotin. Most of our tools have been developed to recognize B cells specific for the spike and the RBD receptor binding domain. So for the future data I will show you in the next slides, you will see mainly these two proteins used. And also very importantly, we have released, and we are going to release even more in the future, also the mutant variants of RBD and spike. How can you use this protein? The protein can be used to study molecular mechanism of SARS-CoV-2 virus, such as the binding of the spike protein to the native ACE2 or fluorine cleavage assays. They can also be used to detect antibodies in serum or plasma of infected or vaccinated individuals. However, one of the most important applications of the SARS-CoV-2 recombinant proteins is the study of the SARS-CoV-2 specific B cells, and specifically the phenotyping and enrichment of this rare population of cells. And this is what I will show you in the next slides. How does the system work? So first of all, you need to create a so-called tetramers using a simple protocol. One streptavidin conjugated to a fluorophore can bind to 4 biotinylated protein, SARS-CoV-2 protein, by a short incubation at room temperature for 15 minutes. Then the tetramers are added to a cell suspension of B cells. And the B cell specific for the BCR, for the spike or the RBD protein, will bind the tetramers via the BCR receptor. Since the tetramers is conjugated via streptavidin to the fluorochrome, it is then possible to detect the cells and quantify them at the flow cytometer. You can check out the exact protocol for the tetramerization at our website. In all our experiments I will show you in the next slides, we all started from a pre-enriched population of B cells using a releasable system of the label called Realism. And specifically here we use Realism CD90 microbit kit, which indeed allows to enrich CD19 positive cells, which are in the blood, the B cells. After the enrichment via magnetic separation, the bits and the labels, so the antibodies on the cell, on the markers, are actually released via the release system, and it's part of the protocol and of the kit. Therefore, what we further use in the experiment are label-free B cells. And this is also very important for 2 reasons. First, because CD19 is not so highly expressed on B cells and tend to be easily saturated by labels. Therefore, freeing up the marker CD19 from any binding helps a better staining for further downstream application, like flow cytometry. The second reason is that if you want to do enrichment via microbits, you need to get rid of the bits on the B cell so that you can further enrich antigen-specific B cell with a second magnetic labeling. It is also possible to use the same system to enrich the SARS-CoV-2 B cells, also using microbits, as I show you, but also via sorting. So I will show you these 2 examples later. Also, very importantly, it is also possible to start directly from PBNC without pre-enrichment. I would strongly recommend it because pre-enrichment helps you to have a much better resolution in the flow cytometry and a higher and better results in the cell separation. So very importantly, we have used two types of tetramer for the same protein. So basically the tetramers we are preparing two different fluorochromes conjugation, one in PE and one in PE-Vio 770. In this way, it's possible to detect the antigen-specific B cells by gating on the diagonal of the dot plot. In this way, the unlabeled cells are excluded. Here you can see the gating strategy by excluding dead cells and doublet, and then by gating on the CD90, which are all the B cells, and then further on the scatters, and then on the two labels via the tetramers. Furthermore, our panels also included anti-IgG, IgA, and IgM antibody that allows also to further phenotype the isotypes and also CD27 to distinguish the memory B cells. And also you can see here that we have tested our system in COVID-19 convalescents versus healthy control. And as expected, we saw that the healthy convalescent show a persistence of B cells after 3 to 6 months after infection. And this was much higher statistically compared to the healthy controls. And this proved that our system is actually very specific for SARS-CoV-2. As mentioned, it's also possible to enrich the cells via sorting using the same panel used for analysis. Here we have used the MAX1 title cell sorter because in this way we could really obtain several advantages that I will show you in the next slide. As you see, we obtain a very high purity of SARS-positive B cells. And on the contrary, of course, in healthy control was not possible to enrich the cells because they were really low. Why to use the MAX1 title in these settings? This is a perfect system to sort in safety fragile and rare cells. It is indeed used in COVID-19 study exactly to sort patient material that can be at risk, give risk to the operator. The microchip of the MAX1 title is disposable, is closed, and is aerosol-free. And this really helps to avoid carryover or risk of infection of the operator. After sorting with the MAX1 title, we have taken the same cells and then add them in culture. And here we use the B-cell expansion kit. That is a specific kit developed to expand polyclonally B cells via CD40 ligand multimer and also IL-4 cytokine. This combination really helped the B cells to expand and then also to start producing antibodies. What we have observed here is that the antigen-specific, so spike-specific B cells in culture expanded nicely after 14 days, and also they were able to produce IgG specific for spike. Basically, after 14 days, we have collected the supernatant and measured with an ELISA-like system the presence of IgG antibodies against the spike. So not only the antigen-specific cell expanded in culture, but they were also able to produce specific antibodies, proving that these cells isolated with our protocol were alive and active and functional and could potentially be used for antibodies generation. However, there is also an alternative to sorting, that is the magnetic enrichment. It is possible to use SARS-CoV-2 B cell magnetic enrichment via using a second labeling with anti-PE microbit and then run the cells through the column. What you collect as positive fraction will be then the antigen-specific B cells. As you can see from the data, in this way you can obtain a very nice 40 times fold enrichment of the population of need. The purity is not as good as with sorting, but it's possible in this way to have a high enrichment of the wished population, which is a very rare population usually, and then you can further do serial dilution to isolate the single cells and single clones. From the 1st of April, it's also possible to collect, to buy all these reagents and also to have a clear protocol in the datasheet by buying the specific SARS-CoV-2 B cell kits. We offer a SARS-CoV-2 B cell analysis kit that includes all the panels of antibody I showed you before, and either the spike protein or the RBD protein. Instead, for the magnetic enrichment, we offer SARS-CoV-2 B cell microarray kit that can also be either for spike or RBD, and is composed of streptavidin, the protein, and also anti-P microbeads. And now let's step into the therapeutic antibody discovery. So in order to develop therapeutic antibodies, for example, for COVID-19 treatment, but also for cancer treatment, many steps have to be followed. First, you need to define a target and validate it. Then you need to discover which antibody binds this target. Then you have to produce the antibody in a stable system. Once they are produced, you want to test them functionally to make sure that they are optimal for their purpose. For example, as I showed before, if they are neutralizing or they can be used to recruit other cells or opsonization or other function. Very often, antibodies are also engineered to make them more stable or to make them more specific and more able to bind the target with high affinity and so on. And finally, you want to select the right candidate that can be used in clinical testing, so you will have to do some preclinical data. And if then the product goes into the clinical trials, you will have a candidate drug that can eventually, after all the phases of clinical trial, become a final pharmaceutical product. In the second part of the webinar, I will therefore focus on the antibody discovery, but invite you also to follow other webinars available in our Miltenyi Biotec database that explain other steps such as the functional analysis of antibody candidates that you can see here. So let's look now about what is the process for antibody discovery. So what is the good antibody candidate and how do you find it? So ideally, you can assume that subjects exposed to a certain pathogen might be able to develop an immune response to it, including specific antibodies able to eliminate pathogens. And you might want to start from these individuals to collect antibodies that could be effective against the pathogen. In COVID-19, for example, this search was done looking into the serum of convalescent individuals able to neutralize the virus and then recover from the disease. Therefore, most of the approaches to identify antibodies suitable for therapy look for natural antibodies present in the body of humans or animals that have been in contact either with the pathogen or with an immunization to that pathogen. However, an unbiased approach is also possible by using libraries of synthetic genes. So the scope of the antibody identification is to determine the sequence of the antibody in order to clone it in an in vitro system able to express the antibody in culture for a mass production. But how do you practically identify the antibody sequence? Antibodies are found in the serum, but it's basically not possible to just isolate the immunoglobulin proteins from the blood and understand the exact sequence just starting from the protein purification, although some advancement has been done in this direction using mass spectrometry. But the only way to properly identify a specific antibody and its sequence is actually to identify the B cells that can produce it, or the plasma cells, and then sequence the genome in order to determine the sequence of the antibodies that they produce. But how can you find and isolate B cells or plasma cells? So let's have a look at the typical development path and compartmentalization of B cells and plasma cells. The B cells develop in several steps, as you can see here. It's quite a complex maturation process. And then they can become either follicular B cells or marginal zone B cells, and they will enter eventually in the lymph node. At this different step of this complex differentiation, they might develop into plasma cells, and the plasma cells are then secreting the antibodies. But where can you find these cells, and how can you really isolate them, and which are the one appropriate for therapeutic antibody discovery? So first of all, you need to see where the cells are located. If you want to work with patients, or in material from human, you cannot imagine to collect it from from certain tissue because it would not be also ethically allowed. It would be much easier to access the cells in the blood. And as you can see here, a lot of the first step of the B cells maturation happen in the bone marrow. Then the cells goes into the blood. Then they go again in the lymph nodes, so they're again not accessible. And in some cases, they are in blood, like in the case of plasmablast, but only for a very short window of time, so between 7 and 14 days after immunization or after contact with the infectious agent. And at one point, they become memory B. They are in the blood and they persist also in the blood for long, as we've seen, for example, SARS-CoV-2, but then eventually enter again in the bone marrow to become long-lived plasma cells. Plasmablasts also can be found in the blood, but also in this very narrow window. So it's actually not very easy to find cells, B cells specific for a certain antigen, unless you really look for memory B cell in the blood. Plasma cells have a very short window in which are present. And also the second problem is that plasma cells, they don't express anymore the BCR on the surface, so they cannot be fished out by using antigens. So that's why most of the people focus on the isolation of memory B cells from blood. Also, after each single step of maturation, B cells improve the binding efficacy to the antigen. Therefore, it's really important to take them at the very end of the development. In this illustration that I've taken from a very informative review that I mentioned here below, you can see how specific antibodies can be identified starting from memory B cells indeed. The driving idea of these approaches is that the B cells need to be isolated in single clones of identical B cells, all producing the same type of antibodies. Once the clones are then separated, the single step is to identify the ones able to produce antibodies specific for the target antigen, and indeed, ideally, functionally active. Finally, it is necessary to sequence the B cell clones to identify the antibody sequence and then express it in a different cellular system for mass production. After the first isolation of B cells, there are mainly three ways to identify single clones. On the left here is the sorting of the single cells in separate wells, and then the culture on feeder cells in order to collect supernatant to test the antibodies of each clone. In the middle, you see the other option, that is to fuse immediately the B cells with an EBV in order to immortalize them and then sort the single cells into wells. After immortalization, the B cells will not need feeder cells anymore, and they could be able to replicate and then stay in the well without problems. On the right side, you see another approach, which is the direct sequencing and cloning. That makes sense, for example, if specific antigen is known and all the sorted cells are antigen specific. This is the case of COVID-19, where spike-specific B cell can be sorted to identify neutralizing antibodies against the spike protein. This approach might not require single-cell sorting and culture, but simply single-cell sequencing, starting from a population of antigen-specific B cells. Here you can see an example of what we offer as product and solution and instrument to support the discovery of therapeutic antibodies. So first, you can start from the preparation of the sample. You can start from blood, but in some cases, like in the case of cancer, you might start from cancer tissue coming either from patient or from animal models. Some users that want to work on human samples, they might wish to start from different type of starting material. And some wish to directly separate the cells from the blood, so in this case, the B cells from blood, and other, they want to further step of PBMC isolation. We have actually solution for every of these solution and also for different type of starting blood material. After the B cell separation, with the respective type of separation systems, the B cells then can be analyzed using flow antibodies and a flow cytometry, like the MAX1. Then the B cells can be cultured for expansion, and here also we offer some media. As I showed you before, we have a B cell expansion kit that works very nicely, and also other type of reagents that can be very useful in B cell activation, for example, TLR ligands and cytokines, and also, as mentioned, the CD40 ligand multimer kit. And finally, another important step that could be part of this workflow is the sorting. As I mentioned before, the MAX1 title cell sorter is very ideal in the case of fragile cells such as B cells, because it's very gentle to the cells compared to normal droplet-based sorters. And also in case of infectious material starting from infectious diseases, a patient infected with infectious agents is ideal because it doesn't carry over samples, it doesn't allow aerosol, and has disposable cartridge. And then finally, once the antibody has been identified, as mentioned before, there are many ways that they can be screened. And this will imply then the use also and the isolation of other type of cells, for example, NK cells for an ADCC assay, or dendritic cells or any other type of antigen-presenting cells for an MLR assay. And for that, as I mentioned before, I will invite you to look into our website and our offer for all the webinars for Miltenyi Biotec that can give you more information on how to set up assay for antibody validation. As you see, I mentioned before, we have seen antibody discovery, and also I mentioned briefly about functional testing of the antibodies. And unfortunately, we are now at the end of this session, but I wish you that you could also look into our different material available either in our website, or also if you contact our sales representative to really investigate further which are the options and solutions that we offer for antibodies discovery and validation. And I'm really happy that you have been part of this webinar. Thank you for your attention.
Thank you, Cristina, for your informative presentation. Joining us today for the live Q&A session is Dr. Suzanne Bietzke, Product Manager for B-Cell Portfolio at Miltenyi Biotec. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Looks like Dr. Andreoni may be having a little bit of a hard time, some technical issues here. So let's see. Susanna, are you available?
Yes, I am available.
Okay, well, as Cristina gets logged in and can figure out the audio, maybe you could answer a few questions for me. Would that be okay?
Yeah, sure, no problem.
Okay, let's see. I will go ahead and ask, which are the variants of concern available?
So we of course have the Wuhan variant, because it's probably likely the most interesting, especially for vaccine studies, because all our vaccines are based on this variants. You know, of course, this is like, very interesting for the B-cell researchers. And then of course we have the spike variant, the Delta variant of the spike protein. So that's what I wanted to say. And then also the UK variant and some other mutations that are not so common. So actually we have quite some things available. So everything that, usually people look into, we have available.
Okay, thank you, Susanna. The next question, do you think that the SARS-CoV-2 pandemic has caused a better general understanding of the immune system? And could other diseases be understood thanks to the SARS-CoV-2 study?
Oh, that's actually a very nice question. So actually, I think this depends a bit on whom you're asking, so whom you're talking about. So I would say, like in general, in the society, I think everybody now is talking about antibodies and B cells. So even our grandparents that never heard of this story before. So this is really, I think, a topic that was really more fashion, and everybody is talking about vaccinations and everything. So this is I think really nice that this moved more into perspective of the general society. And I think, like, from the research point of view, that more and more people are started now to work more, interdisciplinary. So that, like, people before working just on the T cells or a specific topic also now started to look into B cells, and that we started now to look into, what are those both cell types doing? What is, like, the whole immune system doing? So really look into, like, this, how it works together. So this is like really something that I think, really changed. And I think also that we learned like a lot, about, like, how we can fast, like, use the immune system, like, as Christina said in the webinar, how we can use like antibodies also for cancer, how we can, like, work faster. So I think it really pushed the field a bit so that we can really, like, use this, like, disease for, maybe also, like, how we react to future pandemics. I hope, fingers crossed, that nothing is happening, but I think this really is, like, push to the whole field.
All right. Thank you again, Susanna. Looks like Cristina is still having trouble getting connected, and I'll just go ahead and continue to ask questions to you, and then later, if she can get logged back in, perhaps she'll answer them as well, and if you think she might want to add anything.
Yeah,
So,
I think so.
the next question, can you also analyze plasma cells and plasmablasts with the system?
Yeah, this is actually also like a really good question, because obviously, like, all of us know now that plasma cells are those cells of the immune system that produce actually the antibodies in high quantity. So it's like, really crazy. They're like little factories producing the antibodies. And the problem is just with the plasma cells that they just produce the antibodies. So they don't have this B cell receptors on the surface anymore. So you cannot, actually detect if those plasma cells are producing then antibodies against your antigen. So, like, for instance, you're looking into the spike protein, so you cannot really have the plasma cell then being attached to your protein and select it by that and look into it by that. So then you would rather have to go, culture them and look what antibodies they're producing. But of course, we have this subset of plasmablasts that are, like, in between this activated or memory B cells, and then, become differentiate into the plasma cells. This we can still look into because they have still the antibodies, so the B cell receptors, or the antibody that is bound to the surface of the B cells, they still have it. And so we could still look into them and look if they are binding then the spike protein. And then also you could just add to the panel then the CD38 marker, because that's, like, main interesting marker for the plasma cells. And when so you could just, like, easily exchange, like, for instance, the IgM, because this is probably then not so interesting anymore, because that's, like, more an antibody that's produced in a natural, we call it, like, usually natural antibody, and not, it's not so evolved and not so mature. And so usually when you look into plasma cells, you're not interested in that anymore. So you could just, like, exchange this IgM, anti-IgM antibody, APC, against, like, CD38 APC. So this would be, like, easy exchange. So I think that wouldn't be a problem. Even, like, the compensation could be the same. So this is, like, could be done partially.
Thanks again, Susanna. It looks like we have Cristina back with us. So I will ask the next question here. Can you hear me?
Cristina?
Cristina? I think we have her back. Let's see. I will go ahead and ask the next question, and if either of you want to answer, that'd be fine. Whichever way you want to do that. Can one detect or sort antibodies created via naturally acquired or immunized immunity? Is there a distinction?
If there's a distinction, that's actually, like, a really good question. I don't know how you would, like, do. So you can, like, isolate both and, like, like, get your antibody of both systems. And you can, of course, sequence then the genes and then see how maturated they are, so how high the mutations in the V regions. So these are variable regions that usually change when you have something that is really adapted to an antigen. And usually when this is longer process, when you're sick and you have natural exposure, usually this is higher, the regions are higher maturated, higher mutated. So by this you could differentiate, but not really looking onto the antibodies, probably not also to the B cells. So this is really tricky. I would say if you have proper immunization, it would also probably look the same. That's why you get the boost. So tricky. There you would really need to do some distinctive research and do a lot of sequencing and checking the B genes.
Yeah, that's very interesting because B cells and T cells are B cells and T cells. So I'm interested to learn. Yeah, if that works. Okay, let's see. I'm trying to find an. Okay, so the next question: what is the minimum of B cells needed for the cell culture with the improved medium at the moment?
So actually, I don't really know that by heart. I think I have in mind something around 700. But we have a nice protocol that we wrote, so and really specified that on cell numbers, what do you have to pipette and everything like this, really all the details. So it would be the best, maybe you can also send it around later when I think when we compile information, maybe somebody also wants to get some additional information, so then maybe we can also add a link there, because then I think it was around 700. For B cells, going higher is always possible, but just having a few B cells in culture is always a bit tricky, and therefore it's always a bit I'm always asked this question, how low can we go? But I think it was around that cell number that we tested. And for details, please have a look on our homepage. There we have the protocol for the B cell cultivation, and also in the specific case with sorting the cells before. But we also try to send the email then around later.
All right, thank you again, Susanna. Let's see. I'm going to ask if we've got a few more questions here, and hopefully Cristina can get her audio fixed. I think she can hear us, but we can't hear her. So I apologize for any inconvenience. I'm so glad you're here to answer these questions. Okay, next question. Let's see. Is B cell pre-enrichment needed?
Is B cell pre-enrichment needed? Yeah. Needed not, so you can really do it from PBMC. So we tested both, of course, and the huge difference actually is that you're much faster and get, of course, higher purities when you pre-enrich your B cells. So this is really something that is depends a bit on how pure you want to have it. So do you really mind if there are some other not specific B cells in there? So we usually like to be very specific and very pure. So that's why we added the pre-enrichment, and also for people that want to maybe look into different subsets of cells, so not only B cells. So as we said, we also looked into the T cells and used the very same samples, because also in the beginning it was not so easy to get the patient samples, and then we started first to actually isolate B and T cells from the very same donor, from the very same blood sample. And so by that we got most of the information out that was possible. And so I would recommend it, because I think it's always good that you are purer, but if you don't need it, of course, you can just go from your PBMC stock.
All right, great. Thank you again. See, we've got a couple more questions. What is the advantage of tetramers over single protein conjugation?
Oh, yeah. It's one of the favorite, visa questions. So when you have the tetramer, it's of course, bigger. You have like more sites where the antibodies or where the B cells like can bind with the B cell receptor. And that's of course like, the more antigen you have, the better representation is there and the better the B cells are bound there. So that's why you usually like to use the tetramer. So that's actually a very common technique in the B cell field, that you use like this tetramers, then you have like more binding site, because actually usually like, the protein is like otherwise like, really like tiny, and it's not so easy like, to see them like, in the flow analysis. Also like for the isolation, it's like, doesn't work that good. And the more binding sites you have, the higher avidity there is, so the better binding actually is happening. And that's like also like, something, then you have more specific binding there. And that is always like, something that you want to have, so because then you don't have like, unspecific binding and some, like cells that you actually don't want to have like, in your specific culture. So, or like, in your experiment. And that's why this is always recommended, and that's why we also do the double staining, so that you on top of this tetramerization have like also another signal in the cytometer, so that you can really see your specific B cells that are antigen specific, because there's always like such a, low number. We're talking about 0.1 to 0.5% of the B cells, but are already like so few in the whole PBMC fraction, and that's why this is like always like a good option to use that.
All right, thank you again, Suzanne. It looks like we have Cristina back with us, so I'm so pleased.
I'm
Cristina.
It was a setting of the headset. Sorry, it didn't work.
That's okay. I think we're all used to the technical issues, of this pandemic time. So I will go ahead and move on, and you all both feel free to answer as you please. So the next question: can the tetramer system be used also in mouse models?
Okay, Susanne, if you don't mind to jump in, just this question been faced a few times already. So basically, in principle, yes. So the protein, the spike protein or RBD protein, of course, it belongs to the virus, right? And a mouse can mount an immune response, a B cell response, or a T cell response against antigen from a virus, from a foreigner agent that comes into the body. So in principle, yes. In fact, there are models used to study the vaccination in mice. So you can basically take a normal vaccine that is produced now for COVID-19, and then inject it in mice and see immune response. What is not really well established, I think now there are some models coming, but it was never a good system, was to have the mouse as a model for infectious disease of the virus. But in terms of immunity, that's possible. So based on that, basically, you can actually stimulate a B cell with any antigen. And I mean, in a sense, you can stimulate the whole B cell pool in a mammalian with any antigen. And in this way, also, if you have immunized a mouse with a spike protein, then you will be able to detect the B cell specific for that protein using the tetramer system. So if you inject the spike with a vaccine, then you can also detect that. We have not tried this in house, but we know of some vaccine company that tried that. We have some collaborator that did it actually with our tetramer system, and is working quite well. The difficulty is that you have to also really think about the immunity of the mouse, because it's different than the human. The immune response might be slightly different. You cannot get so many cells from the blood. You will probably look at splenocytes. And so the ratio of cells is different, right? You have much more B cells in the spleen. So probably you have to retitrate the system. And of course, also, you cannot use the panel of antibody we suggested, because those are for human. So our kit would not work. You need to redesign the panel for mouse.
Oh, interesting. All right. Thank you, Cristina. The next question here: why are assays for detection of immunity against SARS-CoV-2 not prevalent?
So I think this question, it's very general. I'm not sure if I'm interpreting correct. Maybe the person can write another comment if wants. Of course, measure immunity against SARS-CoV-2 or immunity against the vaccine, you will mainly look at the antibodies production in the blood. That's what a lot of vaccine companies are doing to detect if the vaccine is working. But of course, the cellular immunity is also very important. So far, but the problem is that cellular immunity is more complex to analyze, and honestly also more expensive, because you know to do an ELISA on antibodies in the serum is much easier and cheaper than to analyze cells in the blood. However, we hope also with our system that we have provided something already well standardized and also relatively affordable that can also become eventually a standard procedure in measuring and in monitoring and screening vaccinated or infected individuals in maybe in big studies. So I think the reason why this is not done yet is because there was kind of an understanding initially that antibody response is the only thing that counts, which is not true. Now with many studies, it's been seen that, for example, T-cell response is very important, also memory B. So now more and more studies will probably try to look into the cellular immunity as well, because as we know, it could be that it's not necessary to do so many boosts, like third, fourth dose of vaccine. Maybe is enough to measure the B-cell response and the T-cell response to really know if people are really protected. And to do that, of course, you have to set up assays.
All right, thank you, Cristina. Looks like we have one more question here. Could I apply this antigen-specific lymphocyte technique on cancer-associated complex glycan? Sometimes the real glycan structure is hard to synthesize.
So, Susanne, do you want to add something here? I have some idea, but...
Yeah, I'm also not 100% sure, what is so you mean, so let me rephrase it. So I think the question means if we could do that on isolated antigens, not synthesized ones. This is really tricky because you always have to couple your antigen then to somehow via usually we did it we did it via biotin to PE, because we have to somehow make it visible in the flow cytometer. So this is a bit tricky, but of course you could couple it directly to your glucan. That's is possible. So I think that is a bit tricky, and you would have to really test a bit around, but in theory that should be possible. But as you know, when you don't produce something in a specific way, it's not as reproducible as when you isolate something from fresh material. As I did that for I worked with parasites, so we also did parasite lysates and tried to do some antibody and B cell selection on that, and never worked as good as when you're synthesizing your proteins. So I think this is really tricky.
All right, thank you so much. Looks like that's all we have time for today. Thank you, Cristina. Do you have any final comments for the audience before we go?
Thank you. I'm sorry for the technical mistake problem, but I believe Susanne was the perfect person to answer all of your questions in the beginning. She might have mentioned in one of the first questions that it's really a good opportunity now with the studies of SARS-CoV-2 to investigate more the immune response, especially the B-cell response. And I think this opens a lot of avenues for more studies and more understanding, not just of viral infection, but also of cancer. And the last question was actually indeed very interesting, because this is also where we would like to move in the future, not just for therapeutic antibodies for viral diseases, but also for the cancer research. So we are happy to get in contact with you. I saw there were also a few technical questions maybe we were not able to answer today, but if you want to be in contact with us and get in touch, we will be happy to help you to set up the best system for your study, whether they are for SARS-CoV-2, any other viral disease, or even for cancer. So thank you very much for your participation.
Well, thank you again, Dr. Cristina Conforti Andreoni, for your time today and for your important research. Thank you also to Dr. Suzanne Biepke for answering so many questions. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.